Well-optimized assays are vital for the proper interpretation of environmental DNA data. This protocol describes the necessary design and testing steps to develop such an assay.
In general, environmental DNA is a non-invasive method for detecting a particular species or suite of species. It may reduce costs and be more sensitive compared to conventional survey methods.
This method can aid conservation and wildlife management through the detection of DNA from a species of interest, allowing one to potentially infer that species' presence in the sampled area.
Because of the great sensitivity of quantitative PCR, contamination can happen easily, and negative controls are needed to identify steps at which contamination was introduced.
eDNA is a field littered with jargon and shorthand terms. For those not practiced in molecular biology or qPCR, visual demonstrations can aid the planning of an experimental design from conceptualization to data generation.
Demonstrating the procedure will be Dannise Ruiz Ramos, a biologist from our laboratory, and Trudy Frost, a student laboratory aide.
To begin, search and download sequences from multiple gene regions for species of interest using NCBI's nucleotide database. Select all sequences that match the specifications and select Send to. Choose Complete Record, File, Download format as either GenBank or FASTA, then Create File. These sequences are now saved to the computer. Repeat these steps for all the species of interest. Keep sequences for each gene region in a separate file, as they will be analyzed separately.